Hi there, Happy New Year 2014. This is Mrs. Meek and this is episode number 10, and today we're going to talk about chromosomes and human heredity. All right, so the first thing when we talk about chromosomes is in order to study chromosomes, scientists use what they call karyotype studies. All right, it's where they are able to basically freeze the cell as it's going through metaphase. They stain the chromosomes, all right, and then they're able to match up the chromosomes into homologous pairs. So we know that we have two copies of our chromosomes, one from mom, one from dad, and what they'll do is they'll pair up the two homologous chromosomes by the banding pattern that they see and by the length. All right, at this time, because of all the technology that we have, most of this is done by a computer. All right, so they will take a sample from a patient, and a computer will basically, using an image of the chromosomes, take a picture of the chromosomes after they're stained, and then it sorts them based on the length, which it can get very precise with, and the banding pattern. All right. When we originally did karyotype studies in the 50s and 60s, they would have to be stained, and then a technician, would look at them under a microscope and physically move them using small little needles, move the chromosomes and stuff until they got them to line up. Or they would take a picture of them underneath take a picture of the image on the microscope and then actually cut out the chromosomes and then paste them in order to pairs. All right, so our karyotype studies have come a very long way. Like I said, most of it's computerized now, and we get really, really accurate and good results. So before we start talking too much more about what we can detect using chromosomal studies such as karyotypes, let's look at what a chromosome just consists of. All right, so we've talked about chromosomes, and this is showing you the two sister chromatids for a chromosome that has been copied during interphase. All right, so if you look at this chromosome, there's some key parts that we need to understand. We have these areas that are located at the tip of each sister chromatid, and those are called telomeres. All right, and they are just these protective caps that are in the end. They do not code for any genes. All right, but it just allows them, if something happens, they get bumped, some of that DNA gets broken off, it will pretty much protect the inner coding region of the chromosome. All right, we also know that these telomeres somehow are able to keep track of how many times the chromosomes have been copied. That way they can go back, and if the chromosomes have been copied too many times, that means the cell's been around for a pretty long time, and it may send that cell into apoptosis, which we know is programmed cell death, and our bodies do that intentionally to avoid having too many old cells around that may be mutated or damaged. So these telomeres are involved with aging, and there is even a link with the telomeres not functioning correctly and cancer. So if you remember back to our unit on cancer, we briefly talked about how cancer is caused by cells that are reproducing out of control. So these telomeres, if they're damaged, can cause cells to become cancerous. All right, where the chromosomes pinch together, we call that a centromere, which we've already learned when we've talked about mitosis and meiosis. And then you have the short arm of the chromosome and the long arm of the chromosome. A lot of times they'll refer to these as P being the long arm and Q being the short arm. All right, but those are just some terms that they use when they do chromosomal studies. All right, the most common condition that occurs when chromosomes are responsible for genetic disease or disorder is called non-disjunction. All right, non-disjunction occurs when the homologous chromosomes or the sister chromatids do not separate during meiosis. All right, so during anaphase 1 or anaphase 2 of meiosis, if either of those events do not separate the chromosomes and the homologous pairs of chromosomes correctly, all right, then we can end up with gametes that either have an extra chromosome or one too few chromosomes, so one less chromosome than required. If the offspring ends up with an extra copy of a chromosome, we call that trisomy, meaning 3. If they end up with only one copy of a chromosome, then we call that monosomy. Typically, in most cases, if you get an extra chromosome, it's fatal, and the person experiencing that, they would just experience a miscarriage or spontaneous abortion early on in the pregnancy. There are some very rare situations in which the child will be born with that extra chromosome, and one of these particular situations is trisomy 21. All right, we talked about how in karyotype studies, the chromosomes are sorted by size, 1 being the largest chromosome, number 21 being the smallest, and then the last pair of chromosomes, your sex chromosomes. So trisomy 21 would result in a child with Down syndrome. So chromosome 21, one of the smallest chromosomes in our cells, if we get one extra copy, it results in all the conditions that we associate with a child with Down syndrome. All right, and if you think about someone that you may know that has Down syndrome, they typically have wide set eyes, a pretty flat nose, very distinct facial features. A lot of times they will have short appendages, short fingers. They will also have some decreased visual and mental impairments, as well as organ issues. So a lot of times they end up needing quite a bit of medical treatment, and typically their life expectancy is not as long as a normal, healthy human being. All right, other conditions that can arise from non-disjunction in your sex chromosomes can be things like Turner syndrome, where a female would only have one X chromosome. If you get an extra X chromosome, which is called triple X, all right, that creates a nearly normal female. Most of the time, it's hard to distinguish somebody with an extra X chromosome. Typically, they don't realize they have that extra X chromosome until they try to have children and all of their gametes have been created and they're not balanced. They don't have the right number of chromosomes in the gametes that they produce, so that causes fertility issues. Same thing with a male with an extra X chromosome. Very few physical symptoms, but a lot of times it gets noticed when they are trying to have children. And then XYY, the male is relatively normal, typically will have some increase because the Y chromosome does not have many genes on it. Typically, it will go unnoticed until they try to have children as well. They may have some increase in facial hair, things that are associated with testosterone. So an increase in facial hair growth, that kind of thing, and maybe even athletic ability. But for the most part, it goes unnoticed. Now, if you are missing the X chromosome completely, then that's too big of a chromosome and it causes too many disruptions, and the fetus will never start to develop. The fertilized egg will just undergo spontaneous abortion or a miscarriage. All right, there are several ways that we are able to test the fetus to determine if there are any of these chromosomal abnormalities present. One way is amniocentesis, and that is where they will insert a rather large needle through the mother's belly button to the placenta where the baby is developing, and they will remove some of the fluid around the baby. That fluid has some of the cells of the baby, and they're able to then do karyotype studies on the chromosomes in the nucleus. There's other things that are called chronic villus sampling and fetal blood sampling. In fetal blood sampling, what they will do is they will take a blood sample from the mother, and they can separate out in the mother's blood supply. There will be both her blood cells as well as some of the fetus's blood cells that kind of carry over into the mother's blood supply. And those fetal blood cells can be separated out from the mother's cells, and they can test the karyotype of the developing child. If couples have been identified before they have children with particular genetic defects, oftentimes they will follow up when they do get pregnant to make sure they have a fetal test done. So if they are parents that know that they are at a higher risk for Down syndrome, they can undergo a test shortly after the fetus is developed to determine if that fetus has that extra chromosome. Also, people that are over the age of 40 carry a much greater risk of having a child with Down syndrome. So older couples that wait to have children, so they are older when they have children, once again their gametes have been around for a longer period of time, may have undergone a mutation, they may want to get tested before or have a fetal test done shortly after conception. So there is a huge increase in risk in women over 40 of Down syndrome, because basically those eggs have been around since she was in her mother's womb, and so they've been around 40 plus years. So that's a lot of time that those gametes could have gotten mutated. All right, so here's a short video that just discusses.
Do you know anyone with Down syndrome or Klinefelter syndrome? These are examples of chromosomal disorders, which are due to non-disjunction. When non-disjunction occurs during meiosis I, replicated homologous chromosomes do not separate correctly. One daughter cell retains both replicated chromosomes. The other daughter cell lacks that chromosome. When a haploid sperm fertilizes an egg with an extra chromosome, trisomy occurs. When a sperm cell fertilizes an egg that lacks a chromosome, monosomy results. In this scenario, either a trisomy or a monosomy results. Non-disjunction can also occur during meiosis II. In this scenario, meiosis I occurs normally. When the daughter cells separate during meiosis II, one daughter cell divides normally, but the other daughter cell divides abnormally. One egg lacks a chromosome while the other egg has an extra chromosome. When a haploid sperm cell fertilizes a haploid egg, a normal diploid results. If the sperm cell fertilizes the egg that lacks a chromosome, then monosomy results. If the sperm cell fertilizes the egg with an extra chromosome, then trisomy results. In this scenario, there is a 50% chance of a normal diploid, a 25% chance of a monosomy, and a 25% chance of a trisomy.
All right, guys, so go ahead and bring your notes to class tomorrow.